So my name is Sharon Peacock. I'm a Professor of Microbiology and Public Health at the University of Cambridge. And my talk today is really going to be looking at how we can sequence pathogens to improve human health. Genome sequencing is where we understand the letters of the genetic code. It's a bit like a string of letters in the DNA, or it can be RNA, that encodes all of the information that's required to build that organism, be it a bacterium or a human being. So the genetic code encodes for everything that we become. What we want to do when we sequence the genetic code is really write out, if you like, the letters of the genetic code so we can better understand the differences in genetic code between different bacteria or between one person and another. Genetic sequencing of microorganisms, particularly focused on pathogens, that's microorganisms that can cause disease in humans, is of great importance because if we sequence the genome of a single organism, that can help us, for example, design vaccines against them. And that's what happened with the SARS-CoV-2 virus that caused the pandemic. It can also help us identify parts of the genome that dictate that the organism is resistant to particular treatments. So, for example, in HIV, we sequence parts of the virus to see whether there's any inherent resistance in that virus, and that guides treatment. If we have the genomes of several pathogens of the same species, for example, MRSA, which causes infection in hospital, we can compare the similarity in the entire genomes of one organism and another to see how related they are, and that can help us understand whether, if they're very related, they could be part of an outbreak, or if they're not related at all, that we can exclude an outbreak. So sequencing is also really important in outbreak detection and management. One exciting future application of genome sequencing is metagenomics, where you apply sequencing to populations of bacteria, in particular the gut microbiome, so the population of microbes that exist in our gut. The microorganisms that we all carry in our gut are really important right from the beginning, from when we're born and we acquire these organisms. They can dictate how our immune system develops. They're also associated with the development of particular non-communicable chronic diseases. Understanding how the gut microbiome links to the development of particular chronic diseases is the starting point for understanding how we can use sequencing of the gut microbiome as a risk predictor for these diseases, and also potentially to manipulate the microbiome that we carry so that it reduces the risk of those diseases. Sequencing pathogens is already widely established in routine clinical practice. So we routinely sequence TB, for example, to predict what treatment to give people, and we routinely sequence the HIV virus to really guide treatment. But in order for this to be brought in, for example, to detect and manage outbreaks in hospitals from, for example, multi-drug resistant gram-negative bacteria, there's quite a few things that we need to put in place. A key one of those is having very easy to use analytical tools that manage large amounts of genome data and actually give an answer that's very usable to the end user. For example, to treat a patient with drug A rather than B, or to confirm or refute an outbreak is going on in the hospital. Those very simple messages to people so that they don't need to be expert in informatics themselves in order to be able to use the information. The most exciting part of my career has been the development of the COVID-19 Genomics UK Consortium. That was a consortium that was developed during the pandemic to sequence the SARS-CoV-2 virus. So we use that information to guide, for example, vaccine redesign, to guide interventions in the country and to detect variants. That was instrumental in how we managed the pandemic from start to finish. It was incredibly exciting to have led so-called COG UK to support our response in this country. I'm delighted to have won the Marjory Stephenson Prize. It is a wonderful tribute, but it's not just a tribute for me. Actually, it's a tribute for the many hundreds of people who've contributed to the work that I'll be talking about today. And in particular, in the COVID-19 Genomics UK Consortium, or COG UK, that responded to sequencing the pandemic virus over the last few years. There were 600 volunteers in our consortium, and what it really means to me is as a tribute to our team effort to have actually got that going and helped the country respond to the virus.